For 50 years, the protein folding problem was biology's hardest unsolved puzzle. A single protein could take a lab team months and millions of dollars to decode. Yet, there are roughly 200 million unique proteins driving every function across all known life on Earth. And AI just solved all of them. In October 2024, the Nobel Committee in Stockholm awarded the Nobel Prize in Chemistry to its creator, Demis Hassabis, recognizing a feat that completely shifted the timeline of modern medicine. If you're over 45, this matters to you more than almost any other development in science right now. Why? Because misfolded proteins aren't just a nerdy biochemistry problem. They are one of the primary physical mechanisms behind Alzheimer's, Parkinson's, and a process researchers now call proteome collapse, the specific point at which your body's cellular machinery starts failing from the inside out. In the next 10 minutes, we are bypassing the superficial tech hype. I'm going to show you exactly how this technology works, what the hard data says about its impact on reversing biological decay, and what it means for you that the very first AI-designed drugs targeting these aging pathways are entering human clinical trials right now. Your body is fundamentally a protein machine. Every enzyme that digests your food, every receptor that signals systemic inflammation, every structural component holding your cells together, all proteins. But a protein cannot do its job as a flat string of data. It has to fold into a highly precise, three-dimensional shape to function correctly. The underlying problem is complex. The sequence of a protein is encoded in your DNA, but that sequence is just a blueprint. The actual folded structure determines everything. The exact same sequence, folded slightly differently, can be completely inert, highly functional, or actively toxic to your organs. This is precisely where biological aging enters the picture. As you get older, your cellular quality control systems, specifically the chaperones responsible for refolding or destroying misfolded proteins, become progressively less efficient. This is a hallmark of aging known as proteostasis decline. Modern researchers believe this decline is one of the core primary drivers of human biological aging, not merely a side effect of getting older. When these misfolded proteins evade your body's disposal systems, they accumulate. They clump into toxic aggregates. In the brain, those aggregates manifest as the amyloid plaques and tau tangles associated with cognitive decline. In other vital tissues, they disrupt mitochondrial energy production, trigger chronic low-grade inflammation, and accelerate the cellular aging clock. For decades, drug developers understood this exact mechanism. The bottleneck was simple but devastating. You cannot design a key to target a molecular lock you cannot see. And that is exactly what AlphaFold changed forever. The breakthrough began with Demis Hassabis and his team at DeepMind. They approached protein folding not as an isolated biology problem, but as a massive computational challenge, a benchmark to prove whether artificial intelligence could solve a foundational scientific mystery that human teams could not crack alone. By the time AlphaFold 2 entered global blind testing, its structural predictions achieved an accuracy level that veterans in the field described as a once-in-a-generation milestone. Within a short window, the team released the AlphaFold protein structure database, making over 200 million 3D structures available to global researchers for the first time in human history. This monumental open-source achievement directly led to the 2024 Nobel Prize in Chemistry. It marked the first time the committee recognized work driven primarily by an AI system. The newest iteration, AlphaFold 3, shifts the paradigm entirely. Where earlier versions merely predicted the static shape of an isolated protein. AlphaFold 3 models complex molecular assemblies. It can show you precisely how a protein interacts with a small drug molecule, how it binds to a specific strand of DNA, or how its structure shifts when it encounters an immune system signal. It operates at true atomic precision, the level of resolution where individual atoms and their chemical bonds are completely visible. To understand the scale of this breakthrough, imagine trying to design a highly secure key for a lock you have never seen. Previous computational tools could give you a rough, blurry silhouette of what that lock might look like. AlphaFold 3 shows you the exact position of every single internal tumbler in real time as the key is inserted. This is the foundation of true rational drug design. Instead of screening millions of random chemical compounds in a laboratory, hoping one accidentally binds to the target protein, the traditional, slow, multi-million-dollar approach, researchers can now work backwards from the 3D structure. They can design customized molecules with an exact chemical fit from scratch. Early data from research groups utilizing this exact approach shows significantly higher success rates in identifying viable drug candidates compared to conventional methods. Even in standard longevity model organisms like C. elegans, studies have demonstrated measurable lifespan extension using candidates guided by AlphaFold 3, molecules that likely would never have been discovered using traditional screening. But to evaluate this objectively, we have to look past the mainstream headlines at what the data actually reveals. The legitimate excitement surrounding AlphaFold is deeply warranted, but understanding its real-world limitations is exactly how we separate genuine medical progress from pure speculation. The technology has already drastically accelerated research into complex age-related conditions. For Parkinson's disease, AlphaFold 3 models of alpha-synuclein, the specific protein that misfolds and clumps within neurons, have allowed scientists to identify entirely new classes of stabilizing compounds that were previously completely invisible to structural biology. However, the peer-reviewed scientific community is highly specific about what the tool cannot do. Top researchers point out that AlphaFold still faces significant challenges with intrinsically disordered proteins. These are proteins that do not maintain a fixed, stable shape, and they happen to be disproportionately common in human aging and stress pathways. Furthermore, predicting a physical structure is not identical to predicting its biological function, and the dynamic, chaotic structural changes proteins undergo inside a living aging cell are still incredibly difficult to model perfectly. The most accurate, honest summary from scientists who utilize this tool daily is this: AlphaFold dramatically lowers the cost and time required to generate highly precise medical hypotheses. It does not replace the rigorous physical experimentation required to validate them. Which brings us directly to the real-world human data developing right now. The reason 2026 is the most critical year to watch this space is not because of theoretical software updates. It is because of what is happening in the physical world. Isomorphic Labs, a specialized spinoff focused entirely on translating these AI capabilities into actual medicine, has built a closed-loop platform that goes far beyond simple structural prediction. Their proprietary system fully integrates protein structure, binding affinity, generative chemistry, and therapeutic optimization into a single pipeline. Following massive Series B funding rounds, they have secured deep partnerships with major pharmaceutical entities, including Novartis, Eli Lilly, and Johnson & Johnson. Most importantly, they have confirmed they are on track to dose the very first human patients in clinical trials before the end of 2026, with an initial primary focus on oncology and immune system pathways. This is a massive milestone. Phase 1 human trials represent the initial formal test of whether an entirely AI-designed drug candidate is safe, stable, and tolerable inside a human body. It is the definitive bridge between a digital laboratory model and a physical clinic. What does this mean practically for someone focused on optimizing their biological age? The most immediate, near-term applications of this technology are targeting the precise protein aggregation pathways we discussed earlier, the systems directly implicated in neurodegeneration and systemic inflammation. If even a single one of these AI design candidates produces successful safety and efficacy data over the next 2 to 3 years, the entire economic and operational model for treating age-related disease will permanently shift. And that shift is moving far faster than the public conversation realizes. There is a foundational layer to this story that most mainstream media coverage completely misses. The protein folding problem was never the final goal. It was merely the entry point to understanding and ultimately controlling the molecular machinery of human biology. Building directly on these structural breakthroughs, advanced AI models are now tackling the regulatory genome, where AlphaFold models the physical structure of proteins. New systems are predicting the precise regulatory effects of non-coding DNA variants, the roughly 98% of your genome that does not code for proteins directly, but acts as the master control switchboard determining when, where, and exactly how much of each protein your body manufactures. Longevity researchers believe this regulatory layer of the genome is where the actual programming of biological aging resides. Your underlying DNA sequence does not fundamentally change as you cross from your 30s into your 50s. What changes dramatically is its epigenetic expression, which specific genes are turned on, which are silenced, and to what degree. AI tools are becoming precise enough to map these incredibly complex relationships at scale. The field is entering an era scientists are calling generative biology. This is the transition from merely reading the code of life to actively writing new instructions for it. We're talking about designing entirely novel proteins that do not exist in nature to clear out cellular debris, or identifying the exact regulatory switches required to downregulate systemic inflammation. We are undeniably at the earliest stages of this transition. Mature, long-term clinical data is still years away. But the underlying infrastructure, the complete protein atlas, the automated drug design engines, the mapping of the regulatory genome is no longer a theoretical concept. It is active, it is running, and it is accelerating at a pace that is catching much of the medical establishment completely off guard. The bottom line is this: an artificial intelligence platform has successfully mapped the physical architecture of human biology in a way that was completely impossible just 5 years ago. That mapping is now being actively used to design precise therapeutic agents targeting the exact molecular mechanisms behind cellular decay, neurodegeneration, and biological aging. And the first human clinical trials are officially underway. The science is still early, and translating a flawless digital model into a successful human outcome is never a guarantee. But the biological mechanisms being targeted are entirely real, and the computational tools being deployed against them are genuinely unprecedented. If you value deep dives into the actual peer-reviewed science of longevity without the superficial hype, make sure to subscribe. We analyze the concrete data that mainstream channels completely overlook.